Hi, I'm Miss Haley with the Huntsville-Madison County Public Library. And for today's STEM activity, which corresponds to our Week 1 STEM bag that we are handing out at the libraries this summer, I'm going to show you how to make a cell model using paper plates. For this activity, we will be providing the paper plates and some instructions, but you will also need a pair of scissors, some crayons, and some tape, glue, or a stapler. It does not really matter which one you use. That is just to attach your paper plates to each other. So to start out with, we are going to start by creating our cell wall. So our cell wall, or our cell membrane, is what surrounds the cell and holds everything together. And we are going to color that in. So I'm just going to pick a color, and I'm going to do it on one plate and only around sort of this outside ridge. So we're going to wait on our back of our other plate. So I'm going to pick blue, and I'm going to begin coloring the outside edge of this plate. So this represents our cell membrane, which is the outside wall of our cell. It protects the cell. It holds all of the cell's insides in. It keeps things from getting into the cell that we don't want in the cell. And it just generally helps protect our cell. So you can color this any color you want. The colors are really up to you. We're not going to do anything accurate in terms of color. We just want them all to be different so that we can tell the things apart. So my cell wall today is going to be this dark blue. So now what you need to do is cut out the center. That's why we didn't color the center of this. This is going to be our little window into the cell. And I'm not just going to cut out the center circle. You can if you want. But I want a little bit more room than that. So I'm going to cut all the way up to the point of the little ridges of the plate. So just a little bit more. It's about a 1/2 inch more. So to do this, because we don't want to just cut through the side of our plate, I'm going to fold it in half without actually folding it. So I'm just holding it. I'm not crimping that edge. And I'm going to put my scissors here, and I'm just going to snip the middle. And that way now I have a hole in the middle, and I can slip my scissors through that and cut up to the spot where I want to start cutting around, and turn my scissors. And I'm just going to start working my way around the plate, cutting out a little circle from the inside of it. And obviously most cells don't have a hole in the middle. This is just going to be our little window so that we can look in and peep inside of our cell. So I'm going to set this aside. You do not need this guy anymore. You can throw it away. But I'm going to set aside my cell membrane. If you want to, you can also color the back side of this plate blue, or whatever color you have your cell, but do not color the inside of the plate. So if you're going to color it, only do the back side. You do not have to. I'm not going to, just because I want to move on to the next step of our process here. So the next thing we're going to add is the nucleus. The nucleus is the cell's brain. So it's what tells it what to do. So for this, you're going to need 2 colors. You want them to be somewhat different. You don't want them to be the exact same color, because then it'll be hard to tell them apart. So I'm going to pick this one, which I don't know what color. It looks kind of reddish, but I bet it's actually pink. And then I'm going to do, let's do yellow. That should stand out from each other. So what I'm going to do is I'm going to draw a somewhat large circle. You want to keep it on the inside. So the trick here is your cell membrane, when we attach it, is going to attach like this. So you're not going to be able to see the edge of the plate. So don't draw anything on the edge of the plate. You want to keep it towards the center. But I'm going to draw a circle about the size of a quarter. And then inside that circle, I'm going to put a smaller circle, about the size of your crayon tip or an eraser. It can be a little bit bigger. And then I'm going to color in that smaller circle, and then I'm going to color in the larger circle around it. And this is our nucleus. which is the brain of the cell. It's what tells the cell what to do. It controls the cell, and it keeps it active and doing the things that it's supposed to do. So it's what tells it what to do, tells it what job to do, all that good stuff. So we're going to color in our nucleus. This is also where RNA is made, and it is where DNA lives. So all the DNA of a cell is contained in a nucleus. And then inside the nucleolus, which is the little part, is where the RNA is. And that's where RNA is made from the DNA that is in the rest of the nucleus. So that is our nucleus. Next up, we are going to be adding the endoplasmic reticulum. It's a word, right? There's actually two types of endoplasmic reticulum. There's smooth and there's rough. So we are going to pick the same color for both, and then we're going to get a dark color. So let's see, what color do I want my endoplasmic reticulum to be? I think we'll go for this bright, we'll go for this red-orange. Red-orange I think will be a good color for that. And then, do I have a black? I do. And then I'm going to use black. So the endoplasmic reticulum is kind of squiggly looking. So we're going to draw some sort of curvy lines. It kind of looks like an amoeba. It's not an amoeba. Amoeba is an entirely different thing. But for our endoplasmic reticulum, it's going to be next to the nucleus. So we're going to draw just some squiggly lines that all connect to each other, like that. And then I'm going to do the same thing over here on the other side. This one's going to be a little bigger. And I did go over onto my edge, but that's okay. I can see most of it here. So that is my endoplasmic reticulum. But there are, like I said, two types of endoplasmic reticulum. So there's smooth and then there's rough. And we're going to make the rough, we're going to put little dots on one of them. So the endoplasmic reticulum is what helps the cell process proteins and fats. So this one's going to be our smooth. So this one is going to be our rough. And to make it rough, I'm just going to put little dots along it to symbolize its bumpiness, its rough nature. I'm just doing that with my little black crayon here. Some dots. So now we have our rough endoplasmic reticulum and our smooth endoplasmic reticulum, where the protein of our cell and the lipid fats are going to be processed. Next up is our Golgi apparatus, which looks kind of similar to endoplasmic reticulum, but it's not as connected. So it's more like long ovals rather than a squiggly line. But the Golgi apparatus is what helps the endoplasmic reticulum by storing the products that it makes, and then it uses those to help make the cell do its job. So we're going to use this color, which is yellow-green. It does not have to be this color. These colors are not accurate to life, I will tell you that. And so on, kind of over here, doesn't really super matter where, I'm going to draw my Golgi apparatus. And they are these long, smooth ovals. So you guys can see that. It's like a really stretched out noodle. And there's usually just a couple of them kind of clustered together like that. That's our Golgi apparatus. And you might just want like one single one, like a little round one. Those are Golgi vesicles. So next up is our lysosomes. Lysosomes break off of the Golgi apparatus and help the cell digest. So we're going to draw that. They're just little round sort of dark colored dots. So we're just going to draw some round circles. Oops, I don't want to reuse that color. What color should I use? Let's use this. That's a turquoise color. So these can just go around the cell. And the lysosomes, like I said, help the cell digest. So we're going to draw some circles. One, maybe one over here, and one right here. And just color those in. And those are going to be our lysosomes. We definitely want our cell to be able to digest things. I'm going to set that crayon aside so I don't accidentally use it. And next up is going to be the mitochondria. The mitochondria is often called the powerhouse of the cell. And it is called that because it makes the energy that the cell uses. So it has two cell membranes, kind of like our cell has a cell membrane. The mitochondria has two cell membranes, an outer and an inner. So to draw this, we are going to, I think we're just going to use one color for it. But we are going to draw an oval shape. I'm going to put them right here. So draw an oval. That's going to be our outer cell membrane. For our inner cell membrane, you're going to draw sort of a wavy oval inside. So don't touch the outside edge of your oval because we want it to be our inner cell membrane. But it's going to be wavy. Like that. So, and yours can be bigger than mine if you want. It doesn't have to be the same size. I'm just trying to fit it all on my plate. So this is our mitochondria, which produces our energy for our cell. And next up is going to be our centrioles. Let me find a good color for it. I haven't used orange yet. We'll use orange. For our centrioles, centrioles are always in pairs. There's always going to be two. And they're involved in making fibers that the cell is going to use when it splits. So when the cell reproduces and it splits into two, it will make the fibers that help that process. So those kind of look like Twizzler pull and peels to me. Like if they're hollow inside. So we're going to do a circle of dots because it's kind of a 3D look. So I'm going to do just a little circle of dots like that. And then from those, I'm going to do a little line. And that lines are all going to go in the same direction. Like that. So I'm going to make a second one of those since they always occur in pairs. Don't want to leave my one little centriole hanging out by itself. That's not how that works. So we have two little centrioles making some fibers for our cell to split. Next up is a vacuole. Vacuoles only occur in certain types of cells, not all of them. And it is a fluid and enzyme-filled chamber. So it's just a little chamber surrounded by cell membrane. It has its own little membrane that's filled with fluid. So for this, you can kind of draw whatever shape you want. It's going to be sort of roundish. And I think I'm going to use this brown. So I'm just going to kind of draw a little wonky oval right here. And that's going to be my vacuole. And if you want to, you can label these things, or you can make a sheet of paper with your drawings and the words on them, however you want to do that. You don't have to, though. I'm not labeling mine because I have my instructions, and you're going to have instructions too. You don't really have to label them. You can always go back and kind of look at the instructions and figure out what things are. So next up, I need a few protein filaments in here. Protein filaments are just little shards of protein that help give our cell its structure. So I am going to, I think, use gray for my protein filaments. And those are just lines. So just put some lines. I'm going to make them a little thick because I don't want to accidentally color over them later. And they're just going to go around my cell in a few places. Kind of like sprinkles. Little protein sprinkles in my cell. Those are my protein filaments that are going to give my cell a little bit of structure. And then the very last thing you need to do is pick a color that you have not used, which might be hard because we've used a lot of colors. I'm going to go with this peach color. Oh, excuse me, apricot. And I'm going to color in the whole of my background because that is the cytoplasm. So a cell, these aren't just like floating around in nothingness. Inside the cell membrane is cytoplasm. And that's what all these things sort of float in. So I am just going to color. I kind of like to just color over my other colors. I would pick a pale color for this. That way all your other cell parts will still show up really nicely. But you're going to color it in. Try to color around your mitochondria, not through it like I just did. I got a little excited with my coloring in, my bad. And that is going to represent our cytoplasm, which kind of holds everything in it. And now we have our cell. So to finish off, we need to attach our outer cell membrane to this. And like I said, if you want to, you can color the other side so that your whole cell membrane is colored. I'm just going to use tape today. But you can also use a stapler. You could staple around the edge. You could also use glue, although glue doesn't work as well for this, you guys. I'll be honest. There's just a really thin edge. And so glue has a hard time holding it all in place. So I recommend tape or a stapler. And I'm just going to tape all around. I'm going to put the piece of tape on the edge and then kind of fold it over. And I'm going to try to be neat about it. And that doesn't always work out for me. I can be kind of messy. But this is going to hold our cell membrane together. Normal cells don't need this, but our cell model does. It needs a little help. Go all the way around until you don't have any pieces that feel like they're coming off or pooching or doing anything weird. I need another piece right here. I've got a little bit of a hole. And we have our finished cell. So I'm going to one more time, just before I stop, go over our parts of our cell again. So we have our cell membrane, which is the outside part of our cell that is holding all of our cell together. Then we have our nucleus, which is our cell's brain. The nucleus holds the DNA, and the nucleolus is the small dot, which holds the RNA, which is made from the DNA in the nucleus. Then we have our endoplasmic reticulum, which helps the cell process protein and lipid fats. These are those. So we have smooth endoplasmic reticulum and rough endoplasmic reticulum. Then we have some Golgi apparatus, which is this right here. The Golgi apparatus stores the products made by the endoplasmic reticulum. So whatever is processed from the protein and fat that the ER uses is stored right in here, and it helps it work. Then we have our lysosomes, which are these right here. They kind of break off from the Golgi apparatus, and they help the cell digest things. So those are our lysosomes. We have our mitochondria, the powerhouse of the cell that makes energy for the cell. It uses the products and makes energy. Then we have our centrioles, which are these little guys right here. They might not be showing up too good now, but those are involved in making fibers for when the cell splits. And then we have vacuoles. We only have one in this cell. A vacuole is just a fluid, an enzyme-filled chamber. Then we have our little protein filaments. And last of all, the background is our cytoplasm. We hope you have fun with these guys, and we would love to see what you make. Thank you.